So as we enter into our 75th Independence Day, so being the scientific community, it's our duty to come forward and work for the country's progress. So our seminar is a small attempt in this direction where we are discussing the recent technological advancements in the area of science and technology. So today for this evening session, we have with us Professor Bharat Biswal. He is currently the Distinguished Professor, Department of Biomedical Engineering, New Jersey Institute of Technology. Sir has done his MS in Electrical Engineering from the Michigan Technological University. Thereafter, Sir did his PhD in the Department of Biophysics, which was a joint program between the Biophysics Department at Medical College of Wisconsin and the Biomedical Engineering Department at Marquette University. Sir has also done his postdoctoral fellowship at the Biophysics Research Institute, Medical College of Wisconsin, Milwaukee. Sir's research interests include the calibration of fMRI signal, the resting state functional connectivity, clinical application of fMRI, and the connectivity with particular emphasis on drug addiction, ADHD, depression, and traumatic brain injury studies. Professor Biswal is the intellectual founder of the resting state function connectivity with fMRI time series and is responsible for a paradigm shift in imaging neuroscience. His contributions have been enormous, ranging from a deeper understanding of the endogenous brain activity through fMRI to important strategic initiatives in neuroinformatics and data sharing. His early work established that resting state function connectivity as a way of probing functional brain architectures. His work underlies a substantial proportion of current fMRI studies in the literature. In addition, he has provided some fundamental insights into the neurovascular coupling and the multimodal correlates of endogenous and induced brain responses. He was also the pioneer to apply dynamic causal modeling to resting state fMRI time series and continues in a productive and a biophysically informed way to address key issues in brain imaging. Apart from this groundbreaking research contribution to the system neuroscience, he is the co-founder of FCON 1000 dataset. This is the largest open source dataset for fMRI and is acknowledged as the first substantive example of data sharing of its sort. Sir is also the editor-in-chief of the Journal of Brain Connectivity. Academically, his impact is reflected in the fact that he is the highly cited researcher in neuroscience and behavior. So far, he has authored and co-authored more than 150 articles appearing in peer-reviewed journals. His work has appeared in high-profile journals in psychology, neuroscience, and engineering, which includes the Journal of Neuroscience, Proceeding of National Academy of Sciences, Neuroimage, Cerebral Cortex, Human Brain Mapping, and many more. Besides, he is a much sought speaker who has given more than 60 invited talks. He has been continuously funded from the NIH. He has also received funding from NSF, New Jersey Commission on Spinal Cord Injuries, Swiss National Science Foundation, Autism Research, and many more. In the recent years, he has taken a major leadership role in the International Human Connectome Project, which is rapidly accelerating brain research. He is one of the main organizers of the prominent international conferences on brain connectivity and has initiated international exchange programs for researchers in the field, leading to a major multi-site initiatives. So the Neurocomputing Lab feels elated to have you as a speaker for this session. With these words, now I would request Professor Biswal for his talk.
Um, Thank you, Dr. Kaur, for a very long and very generous introduction. I would like to thank the organizers, especially Dr. Gandhi, for giving me an opportunity to talk on my research topic. So, can I share my screen? Okay. Yeah. Can you see my screen?
Yes, sir, it's visible.
Okay, great. Good. So perhaps you could okay. Just okay. Yeah. Um, so if you have any questions, perhaps you can ask me at the end or put it on the chat. Um, so, as Dr. Kaut mentioned, my name is Bharat Biswal, and my talk is about functional MRI and brain connectivity. So, this is a picture from a very famous project called the Human Visible Project. This was done maybe 20, 25 years ago, where a person, maybe I should first say that, so this is an image of the person using CAT scan, using PET imaging, and using MRI. And this is an actual picture of the person's brain. So this person was on death row, and before he was to be executed, he decided to do something good for the society, and he donated his body for science. So basically, what was done was a number of different modality was used to image this person at, um, at the latest technology which was available at that time. And then after the person was executed, um, digital pictures of the person was obtained. Um, and then, as you can see here, again, I just want to emphasize this is about 25 years ago. And this is a comparison between actual brain and an MRI picture. And with improvement in technology, I would say that even the CAT scan looks similar to the, like, not the same resolution, but there's been a lot of improvement in technology where we can get even better resolution, spatial resolution, and we can acquire it in much shorter period of time. And that has been the power of medical imaging. And while there has been a large number of work which has looked at very high structural imaging of the brain, mammography, prostate, and many other organ in humans as well as in animal imaging, there has been a lot of focus in recent years on understanding the function. So, as a simple example, one can think that while this is a structural image of a Simpson, this is a functional image where what we can see is not just the areas, but we can also see what areas are involved with what task, like which regions are involved in sleep, which region are involved for vision, and so on. And this is one of the first ideas of doing a brain imaging. And it was done by an Italian physiologist by the name of Angelo Mosso. And his idea was that if you place a person on a seesaw and then ask them to do complex tasks, like compute complex tasks, then if they perform the task and the task is difficult task, then there will be more blood flow to the brain and the seesaw will tilt. If it didn't tilt, it meant that the task they were doing was not very difficult. So while it may now seem very naive way to think, but indeed, this is the way most brain imaging experiments work. And this is the underlying mechanism that when we perform a task, different regions in the brain which are associated with the task, there is more blood flow to that. And that is what we use in fMRI. So in fMRI, what is done is maybe the analogy is between a picture taken using your smartphone versus making a movie using your smartphone. So with a picture taken, you get very high resolution features. You can see distinct anatomical location, while with picture you get each frame has low resolution. However, when you make it into a movie, you can see interesting feature which may not be visible, like a child running or someone smiling, or someone slapping someone at the Oscars. Right. So in MRI, so this one is a high resolution image with 1 millimeter by 1 millimeter, which takes a few minutes to collect, about 3 to 5 minutes, while this is a functional MRI. And in this case, it is the same image of the brain, excepting because it's a 3 millimeter by 3 millimeter, you can see the overall shape, some of the structure also, but the finer details are not as clear. However, in this case, what we have done is that we are collecting data every second or every 2 seconds. Now we can even collect these at better spatial resolution as well as better temporal resolution. And just as Angelo Mosso had described, in our case, what we can do is we use a mechanism called BOLD, which stands for blood oxygenation level dependent signal. And so using this, what is done is that when we perform a task, let's say we tap our fingers or we watch a movie, there is increased neuronal activity in very specific regions of the brain, right? If we tap our fingers, then there is more activity in the sensory motor cortex. If we watch a movie, there is more activity in the visual cortex. And when there is more activity, it leads to more oxygen consumption. And when there is more oxygen consumption in the brain, it sends out a signal saying that it needs more oxygen. And the way it gets more oxygen is through increased blood flow. And when there is increased blood flow, there is more oxygenated red blood cells. And that is what gives rise to an increase in fMRI signal. Right? So while Angelo Mosso didn't know about BOLD mechanism, what he had hypothesized was the increased blood flow. And that part he was correct. This is the first fMRI, or I should say one of the first fMRI experiment where a person was shown a flashing checkerboard for 60 seconds. So the first 60 seconds was blank screen. Then the next 35 seconds or 25 seconds was, the next 60 seconds was flashing checkerboard. And then again, the next 60 seconds was blank screen. And the following 60 seconds was again flashing checkerboard. And what you can see is when you look at a region from the visual cortex, this is how the signal is. This is during when the blank screen, there is signal activity. When they are shown that when there is blank screen, the signal goes down and goes up again, right? And if you look carefully, this is during blank screen. When the visual stimulus is shown, there is more activity here. Again, during blank screen, it goes back. And then again, during the second period, the signal goes up again. And because fMRI does not have any side effect, there has been an exponential growth in the number of publication and in the number of clinical use. And there's been a lot of study that has been done to study brain function. And the basic idea here is that in an MRI machine, when you place a person, what you also have them do is you can make them watch some movie, you can make them listen to some text, conversation, or music. You can also make them perform, like press some buttons. Basically, you are doing what has happened is that inside the magnet we have almost like a psychological experimental setup where person who is being scanned can perform specific tasks, and then we can detect these tasks and look at which regions of the brain these are happening, right? So just as a brief background for NMR, which stands for nuclear magnetic resonance, it was first demonstrated by Felix Bloch and Ed Purcell, for which they won the Nobel Prize in 1952, showing that atoms had nuclear magnetic resonance. And it was then from NMR, the technology was changed such that we could also gather an image. And because like nuclear has a negative connotation, it was changed to MRI. So the MRI also, there's been some controversy by the Madian, who had a patent about tumor detection, but Paul Lauterbur first demonstrated that using NMR you could generate image. And then because the technology was so impressive, within a few years, the clinical MRI was built and marketed. And then Peter Mansfield proposed echoplanar imaging, the images for functional MRI that I showed. And Seiji Ogawa in 1990 observed the T2 star, or the bold signal, showing that the blood vessels became more visible as blood oxygen decreased, enhancing the contrast. Jack Bellevue observed functional image using a contrast, and then Ogawa and Ken Kwong used the bold signal to image the brain, right? So this is Seiji Ogawa's bold signal, where this is showing with oxygenated blood cells, there is no loss of signal. With deoxygenated, there is signal loss, and you can see some of these vessels, right? So when someone performs a task and there is increased blood flow, this is the reason why there is an increase in signal intensity, because as there is more oxygenated blood, there is less signal loss, therefore the signal goes up. This is again just showing the red blood cells, the red oxygenated red blood cells, and these are deoxygenated red blood cells, the ones in purple. So during activation, because there is more red blood cells, there is more oxygenated red blood cells, and therefore an increased in fMRI signal. There has been a seminal study by Nico Logothetis, where he showed that the activation that we see in fMRI reflects the local field potential slightly more than multi-unit recording. So while what we observe with bold signal is not 100% neuronal activity, but it is a mixture of neuronal activity and local field potential. Nonetheless, because this is a completely non-invasive procedure, that is why there's been a lot of study since then to map the human brain. And this is another study where in a tumor case, where the skull was open, and then electrophysiology study was done to look at hand representation and face representation. And then this was, before doing this, it was done using fMRI. And there was a good concordance between what the hand representation was and the fMRI, which region it showed. And again, this is another slide showing the like a comparative measure between the different modalities. So MEG and EEG here, they have very good temporal resolution. You can collect 100 data points every second in MEG or EEG, no problem. However, the spatial resolution is fairly poor. Similarly, for PET imaging, you get slightly lower temporal and spatial resolution, right? While fMRI has a nice sweet spot where the temporal resolution is good and the spatial resolution is also good, right? So this is just showing that if we collect like a person performs a task which has two conditions, then how do we analyze the data? And the simplest way is we can find a specific location in the brain, let's say something which is of interest or where we hypothesize the brain activation would be. Then we look at, is there any difference between the two conditions? Right? And if there is, then we note it and then go to the next location, and then we do it for every voxel in the brain, and then basically show that which regions of the brain are activated. Okay. And here is another simple example just to get you familiarized with this. So this is showing like a person is being shown a blank screen, intact object, again a blank screen, and then a scrambled object, and then this is then blank screen. And this is repeated every 8 seconds. Right? Blank screen, intact object, blank screen, scrambled object. So in this case, the idea is that when you see, let's say, a green pepper versus when you see a scrambled object, would you see a difference? While we can all agree that, there will be a difference compared to the blank screen, but like what causes the difference between an intact object and a scrambled object when the information is the same, right? So, and then when one analyzes the data, what we see is that during blank screen, the signal goes down. However, during scrambled object, the signal also goes up, but not as much. Again, suggesting that the color, the texture, and the brightness all play a role in the activation. Again, and these are ways in which we can identify which regions are responsible for what task, right? And again, because the fMRI or the data that we are analyzing is a three-dimensional data set, we can display it in many different ways, right? We can find the location and then um, look at the signal intensity, look at the overall intensity, or even display it using different ways. And one can use the basic idea that I presented to ask very specific question. Here, what is being done is not just looking at flashing checkerboard. Here we are asking very specific question. Which regions, for example, in the brain are associated with objects? Which regions in the brain are associated with faces? Which regions in the brain are associated with places, right? So all these three, indeed, we would be presenting visual stimulus. By using these kind of study, we can find out or determine that, okay, the lateral occipital area is associated with object, the parahippocampal place area is associated with places, and the fusiform face area with faces, right? These are, I think, as Dr. Kaur mentioned, these are some of the topics that I will be covering in a little more detail. So just to recapitulate, the task-based fMRI, in task-based, you have two conditions, the rest condition and activation condition. And when one performs, there is an increased activity, right? There is baseline, there is increased activity, there is baseline, then there is increased activity. And as I mentioned, there are many different ways to analyze. And the basic idea is we compute voxel-wise statistics and then use some threshold to identify, like find reliable activation pattern. Then we display it on the brain and say that when you do this task, these regions are associated. Right? So during my PhD, I was more interested, or I became interested to look at what was happening to the brain while we were resting. And this is just showing a diagram. This is the noise outside the brain. So it's quite noisy. Also, the outside the brain, the noise is much smaller than the noise that we see inside the brain. This is from gray matter region, from the visual cortex. This is the heart rate. This is the first harmonic of the heart rate. This is the respiration. And then we see this 1/f parameter or characteristic. And so this is how a time series during rest looks like. And you can see this fluctuation corresponding to the respiration signal. But if you look, there is this low frequency fluctuation. So what we did was we analyzed the resting state data identical to the way we analyzed task activation data. So what we did was we took the resting state data and correlated with every voxel in the brain. And what we saw is that this is when a subject is tapping their finger and identifying the sensory motor cortex. This is when the subject is at rest, and we are analyzing just using the resting information, right? So what we showed was that you didn't even have to tap your finger to identify which one is the sensory motor region. Okay. However, in case there are some, I think there are a few students, so I should say that my paper got rejected 4 times from different journals, because it's not that people disagreed, they just did not believe it. However, after it was published, um, we had used the term resting state functional connectivity because Carl Friston had defined the term functional connectivity as correlation between two time location. Then what we did was we analyzed, we asked a large number of groups who had done work on resting state to share the data. So we had more than 1,000 subjects. These were from around 30 different research sites, like 17 different countries. And obviously the race was different. The scanner was different, the manufacturer was different. Some was collected at 1.5 Tesla, some was collected at 3 Tesla, 4 Tesla, and 7 Tesla. And when we analyzed this, what we could see was that, again, this is the visual cortex, this is the sensory motor cortex. Again, suggesting that the brain is organized into functionally related regions, which we can identify during task. So while task activation, I should say, has a lot of application and importance, what this showed was that in cases where someone could not do a task or time was limited, you could use resting state and map out the brain. In this case, because we had so many subjects, we could look at differences between male and female. We could also ask the question, what happens with aging? Which regions people become better with age, and which regions people become weaker with age. And I think, as Dr. Kaur had mentioned, that all the data that was used to generate these paper, including how we analyzed it, all the scripts and the data is available on FCON. So anyone who is interested can simply download the data and play with the data. So as I mentioned, like fMRI activation has many applications, but in some clinical application, the subject may not be able to even perform. Like in cases of infants, like what kind of tasks would you ask them to do? Or if someone had Alzheimer's disease, what kind of tasks would you have them do? Right? So that is where resting state comes in. So this is just showing an example in children with ADHD. So these are 5 different networks generated from one resting state scan. These are children, healthy, typically developing children, and these are adults, healthy adults, right? And then we can immediately see which region there are differences between ADHD and kids, which region there is difference between, let's say, healthy control, like adults versus children, right? And this is some work um, which was done in collaboration with Dr. Gandhi at IIT. Like this is data collected from COVID subjects showing which region there is uh, increased activity in COVID subjects um, versus healthy control for different, like, again, this is just using the resting state scan. This is another example where a patient or four patients with brain tumor are shown. And in these cases, the activation um, task was finger tapping was done for activation, and then this is using resting state, and then we are showing overlap, right? And again, I want to emphasize that in cases like this, if you can perform a task, one should perform, but in cases where it cannot be done, the resting state could be done. Like in some cases, because someone has a tumor or a stroke, even if they wanted to tap their finger, they may not be able to tap their finger. And that is where resting state would be helpful. In another application, there's been a lot of application in Alzheimer's and schizophrenia. This is one of the first studies looking at Alzheimer's and showing how the connectivity pattern in Alzheimer's is lower compared to healthy control. What is shown here is showing that that Alzheimer's, frontotemporal dementia, and there are other disorders, which are all neurodegenerative disorders, which become like progressively worse with time. And these are the same kind of disorders. So what was done was the question was asked that for these 5 different kinds of disorder, though they have a similar origin, if you will, that would the same regions be associated, or could resting state brain connectivity used to separate them? And this is showing how, with using resting state, one can see differences in structural pattern. Like while for Alzheimer's, we can see these regions. For frontotemporal dementia, we see different pattern, right? Again, suggesting that these different disorders have distinct spatial differences. So this method could be used because it has good specificity in addition to sensitivity. This is showing people who are at risk of Alzheimer's, right? And how that pattern is different and how it can be used to see progression and even tell if someone is getting worse. So then the question was, does the resting state have a neuronal basis? As I mentioned, like there was a lot of resistance initially with the resting state because people thought there may be some kind of activation or some artifact. So here I want to discuss a little bit about the underlying mechanism. And what I want to say is that the fMRI signal that we measure is a convolution, or it uses neuronal activity and underlying hemodynamics, and we get the fMRI signal. So both these factors play a role. And so could we modulate each of these factors to see how the fMRI signal changes? And this is showing where the subject, this is task activation, this is resting state, and then the subjects were made to breathe 5% carbon dioxide, which increases blood flow in the brain by about 70%. And what would happen in that? So one of the underlying hypotheses we have is that these fluctuations that we see is formed due to obtain a good balance between blood flow and metabolism. And when there is an excessive increase in blood flow, the coupling between metabolism and blood flow is disrupted. So the connectivity pattern is disrupted. The subjects then again breathed room air, and several minutes later, another scan was done, and we can see the connectivity pattern back, right? Suggesting these are physiological origin. This is another study where with the anesthesia, what was shown was that there was a great increase in the visual cortex activation, suggesting that there was this increase. This is a different kind of anesthesia where during a wake, we can see bilateral connectivity. With light anesthesia, we can see connectivity on one side, but not connectivity between left and right. So we could see either left or either right, but not between left and right. But with deep anesthesia, both within and between connectivity is affected. In the next 15 to 20 minutes, I'll try to cover some work linking the connectivity between task activation during rest, and then between higher frequency. As with technology, as we collect more data at a faster rate, how can we use? Then we will cover dynamic connectivity and white matter functional connectivity. So one question was that the task activation map that we get and the map we get during rest, what is the significance between them? Are they showing the same thing, or are they showing information? Hello?
Yes, sir, you are audible, sir.
Okay, somehow it kicked me out. So let me try to just go back to my presentation. Oh, okay. Can you see my screen again?
Yes, sir, we can see, sir.
Okay, great. Okay. Also, I have like 15 minutes now. Or am I doing okay on time?
So actually, we are having session timing till 6:30. It's all up to you, sir. The way you want to go ahead, sir.
Okay, no problem. Thank you. Okay, so what we did was we wanted to find out for every location in the brain, which other location had been associated with task. So this is a location of the default mode. So this will find out using this NeuroSim database, we can identify which other regions are associated with this region. So we can identify the co-localization of different regions in the brain. And then we identified 140 regions and then looked at these connectivity pattern. And then we did the same thing with rest. And what is shown here is the co-activation network, right? So what is shown here is for every task that we do, for every region that has been activated, what other regions have been activated, and this is shown here. And this is the connectivity pattern from the resting state correlation matrix. And when we plot one versus the other, we can see that there is good correlation between them. And so one way to think is that the correlation matrix from resting state is like a superset on which task activation is built. But we also wanted to see what is the difference between these patterns. So we did graph theory, and what we can see here is that this is the random network. This is the resting state correlation network, and this is the co-activation network. So in correlation network, you can see different regions there are different networks which talk to each other. But in co-activation network, what we see is larger region which are highly connected between them, right? So this is the task activation, and this is a random network, right? And further looking at this, what we find is that the thalamus is associated with the task activation, while in activation, when in resting state, it is less so. And so when we compare task activation with resting state, the task activation has higher global efficiency, suggesting that they are more, the strength is higher, lower clustering coefficient, lower modality, and higher degree in thalamus. For resting state, there is low global efficiency, high mean clustering coefficient, higher modularity, and there is more activation in the temporal lobe. As I had mentioned in the beginning, that for EEG data or MEG data, we can collect up to 100 data point or even 1,000 data point, while with fMRI, most of the data is very slow fluctuations, right? Up to 0.1 hertz. And now that we can collect more number of data points, maybe 3 data points, like up to 500 millisecond, each volume of data, could we look at higher frequency data? Okay. So this is now looking at task activation at different ranges, right? So this is the very low frequency called slow 5, slow 4, slow 3, slow 2, and slow 1, right? And beyond this is the, delta, alpha, beta waves. We are not there yet, but we wanted to see if there was any fluctuations beyond the 0.1. And again, it suggests that most of the signal is within slow 3, but when you go higher, we see some pattern, but they are not as strong as what we see in the low frequency one. Okay. And with evolving technology, we hope that these questions can be answered, and hope that maybe someday one can collect data where the sampling rate will be similar to EEG. Next is modulation, right? And while I showed you connectivity, the connectivity as such is like varies across time. And while researchers have speculated that fluctuation, what we wanted to do is look at what factors could contribute to dysfunctional connectivity. And so before, people have shown that if two regions have time varying connectivity, so the question we are asking is that if there is variable connectivity between A and B, could it be possible that is the region C which is affecting these connectivity, right? So the question we asked was that are modulatory interaction present in resting state? And modulatory interaction between the default mode network and task positive network, and whether modulatory interaction are modulated by task demand, right? So the question we are asking is that could a region affect two other regions which are connected? The second is that could a network be affecting two other networks? And the third question is, could two regions be also affected by task activation? Right? So this is showing regions from two different data sets. This is the discovery sample where we found that for again, these are the default mode regions, and which regions are contributing to their connectivity pattern, right? So these two are highly connected regions, like these two regions, these two green dots. So we wanted to find out which regions are affecting their correlation. And the one in blue are the region which are negatively affecting. There is the region in red which is positively affecting, right? And on the replication data set from a different data, we see very similar pattern, right? On an independent data suggests that indeed within a network, there are regions which are influencing each other. Then we use the same analysis procedure to look at interaction between two different networks, right? So there is the default mode network is one region and the salience network. So these are the regions which seem to affect their connectivity pattern. Again, suggesting that in many clinical applications like schizophrenia, these regions could be involved in this mismatch between the two different regions. Third, as I mentioned, that between eyes open and eyes closed, the physiological fluctuation, the alpha signal is affected. And this is showing, at least in terms of FI, which region there is a difference. Again, these are all work in progress, suggesting that there is a lot of interaction between the different brain regions, and it's not just as simple as doing a task and looking at the response. There's a lot of connection and talk between the different regions. So for the next one, we wanted to ask the question, if during rest there is such widespread activity, could it be happening that similar kind of activity be happening during task activation? So we looked at specific tasks and looked at two different analysis methods called PPI, which I showed you before, and beta series. Right? Sorry, this is the only equations I have. So with functional connectivity, what we are doing is we are looking at the correlation between X and Y, and this tells us how the strength of the connectivity is. In task activation, you have the stimulus and the response, and that tells you how the task response is. In here, what we are doing is we are adding both and looking at the interaction terms, right? So we used this task, which is available from the Human Connectome project, a number of different kinds of tasks. And this is the response for emotion task. This is the task response. For the gambling task, this is where the activation is. Like the red is more activation, the dark blue is negative activation. And this is what has been shown. And so what we did was we looked at which regions there is interactions, right? Which are the activated region. And so what we see is a large number of area where there is activation, and it seems to change with task. This is the language region, this is the social cognition task, this is the working memory task, this is the stop signal task. And again, suggesting that all these tasks, there is a lot of interactions between them, and based on the task and the level of difficulty of the task, there is interaction between these responses. Right? So by analyzing a whole brain task modulated connectivity, we demonstrated that task modulated extend far beyond regions that are activated during a task. This suggests a broader involvement of regions in different tasks in terms of modulated functional integration, and control condition can enable us to better infer the function of the task. So there's a lot of work to be done. So if I still have 30 minutes, can I just show you one more work, which would take maybe 5 to 7 minutes?
So definitely, sir, actually the participants are like, they are students from different backgrounds, and they will love to hear from you, sir.
Okay, all right, so just give me 1 second. And in the meantime, if someone can, like, if you have any, if anyone has any questions, they can just post it on the chat, and then I'll try my best to answer.
So actually, we have some questions. So if
Okay.
it is comfortable with you, I can just read them out.
Okay, no problem. No problem.
Yeah. So this is one of the question from one of the participants, Kritika.
Okay.
She wanted to ask, what clinical inferences can we make from the resting state functional connectivity? For example, if we get functional connectivity alterations in some clinical population as compared to the healthy controls, how can we make the clinical correlations with it? And also, if you can give some examples from the visual or eye disorders, it would be great, sir.
Okay. All right. That is a great question. And so for example, let's say I'll give you an example from amblyopia, for example. Or actually, let me even go to even simpler one. So let's say someone was born blind versus someone became blind as an adult. So then the question would be, which one do you think would they show differences in their connectivity pattern of their visual cortex? And the answer is perhaps not that surprising. So in people who became blind as an adult, the visual cortex is still intact because they became blind later in life. So at the early stages of their life, the visual cortex was formed, and then it may not be getting much input, but it is there. In the case of people who became congenitally, who were born blind, right? In their case, the response or the visual cortex pattern has typically been taken over by other regions. So the visual cortex is much smaller, if you will. And so there is this, like, sometimes they say that, people who don't have one sense have heightened senses in the other area. May be true that, because there is other region which has taken over the visual cortex, so the other region have become more stronger in those regions. Of course, these kind of studies are very difficult to do, at least in US. But there's several studies like from China where they have done studies like this. And in terms of amblyopia, like would be another example where we have scanned people who have amblyopia and then looked at the visual cortex and how it has been affected. Um, in terms of the clinical relevance of like a clinical disorder in resting state and how it reflects the underlying symptoms, I would say a lot of work has been done in schizophrenia, particularly, where there's like in US, it has been very well studied, and there's a bunch of clinical measures. Like there's a go/no-go task which is used about inhibition, and it has been shown that in schizophrenia, some of the resting state measures correlates with these inhibition tasks. But again, I think, Kanika, there's a lot of work to be done. And initially, a lot of work was done to show differences between two conditions. And now people are looking at, does it correlate to clinical measures? That's one question people are asking. The second question people are asking is perhaps even more difficult, and that is that, if we have, let's say, if someone gave me one data set, would I be able to tell what this person is suffering from? That is this person ADHD? Is this person schizophrenia, or is this person Alzheimer's? So we are not at that stage yet, but hopefully as more people are collecting data, it will be done hopefully sooner than later. Okay, so let me just, I won't take more than, again, I can try to answer, and if, should I just try to answer the other questions? And if there is still time left, then I will cover the white matter.
So we can go ahead with the longer timings as well.
Okay.
And so if it is comfortable with you, we can just have a couple of quick questions and then we can restart from your presentation again, sir.
Okay, no problem.
Yeah. So we have 2 or 3 small questions.
Okay.
So in one application you discussed about the brain tumors, and there were some activations were shown nearly, like maybe near the tumorous region probably. So just wanted to know, like, what kind of additional information is provided by such kind of analysis, and whether that would be helpful for the differential diagnosis.
Right, for tumor, it's a very good point, right? But as I said, for tumor, the question, like if someone could tap their fingers, right, to activate the sensory motor cortex, in the fMRI experiment, you typically would have to tap your fingers and then get a response. However, in some cases, like because in fMRI, the time is limited and the analysis is typically not done in real time, although that is changing. So sometime we would collect the data and then analyze it after the patient has been taken out of the scanner. So in that case, resting state was useful. Also, in tumor, the tumor is typically surrounded by healthy tissue. And sometimes having a number of different tasks helps in a sense that it will at least tell you that what is the role of the tissue surrounding the tumor. So for the neurosurgeon, it may provide them with a path of how to do the surgery. I hope I answered the question.
Yeah. Thank you so much, sir. It was indeed very informative. Participants, if you have any questions, we'll unmute you. So till then, we have two more questions in the chat box by Kritika again.
Okay.
So Kritika wanted to ask, like, what additional information does the task-based functional connectivity give over the resting state functional connectivity?
Oh, that's a very good question, Kritika. So I'll try to answer in terms of resting state, and then try to answer perhaps just from someone who thinks of task activation as a separate entity. So to me, resting state is mostly like a network of interaction between different regions. When you perform a task, you're basically changing the connection between those regions and making them very strong, and that comes up. So just to reiterate, as I showed you for the task connectome or even the PPI work, there's a lot of connection between the different regions when we during rest. When we do the task, let's say finger tapping, then the left and the right motor cortex, the connection is much higher, both within and between them. So that is maybe one way to think of task activation, right? That it is a subset of the resting state. However, in terms so that is my interpretation. And people who are doing resting state, that is their interpretation. But people who are simply doing task activation, for them, based on the task, there is a previous literature, anatomical finding, and therefore they can just focus on those regions, right? So for task activation, for example, finger tapping, it has been established in animal models that there is left sensory motor cortex, right sensory motor cortex. There is another region called supplementary motor region. There is cerebellum. So these are the large 4 regions. And therefore, maybe it's just worth focusing on those regions. So I don't know which one is right, which one is wrong. I'm, of course, biased about resting state, but I think even people who are doing task are beginning to think that maybe there are other regions which are involved, which may not play as strong a role.
So I hope we can take one more question if
okay.
you are.
No problem.
So the other question is, what do you think would be the next breakthrough in the field of resting state functional MRI, perhaps in terms of the method or analysis?
I think in resting state, what would be useful would be to detect to perform a reliable single subject analysis. And I think that would open a number of new avenues if it could be done. Currently, most of the work is looking at group level differences. If we could, if anyone could come up with an analysis method which is reliable and robust, which can do single subject analysis, I think the field would become even like resting state would become much more useful than task activation, in my opinion. But I could say the same thing about task activation also, that even for task activation, if analysis method could be developed to do single subject analysis, that would become quite useful. So that would be one area. The other area would be trying to link gene information or behavior information. And that has been a challenge because some for obvious reason and some for technical reason, right? So all the like genetic studies which are done, especially involving human, are done on like 100,000 or so subject. You cannot get 100,000. Well, you can get 100,000 subjects, but it's not very easy to collect all the information from using a single scanner or single country to do this large study. So, so there's been there's some attempt which has been done. So I think because eventually it would be good to link brain and behavior. So I would say those are the two ones which I think of, but you know, as many of you may realize that science doesn't go in progression. Like it may go in step by step, and then there is a quantum shift, right? So let's see.
So these were all the questions which the participants had to ask. So we can go ahead with your presentation, sir, if time.
Okay.
Yeah.
Okay. Thank you. Right. So for the next 10 minutes, I'll talk about, so what are the assumptions that we have made in resting state and task analysis? Assumption is that most of the information that we see is in the gray matter. And white matter signal have usually been considered to be noise and ignored, right? And at the beginning of my talk, I had mentioned that how in task activation, people had assumed the resting state to be noise and ignored. So just like looking back, we realized that we had indeed done something similar, that is just focus on the gray matter and ignore the white matter. But in the brain, the gray matter and white matter is about 50-50. So 50% of the brain consists of gray matter and 50% consists of white matter. And there's been some work that has been done. And there was a study from Vanderbilt, from John Gore's lab, showing that even in the white matter, when a person was doing different tasks, based on the load of the task, the intensity in the white matter was changing. In another study, by focusing on the white matter, Michael Peer and colleagues could identify several regions in the white matter. So what we did was the largest white matter region is called the corpus callosum, right? And so this is the corpus callosum. So what we did was we took every voxel in the corpus callosum, which is in the white matter, and correlated with every voxel in the brain, right? So for this, we used human connectome subject data, and there were about 110 subjects, and this data was collected at 2 time points, right? So we analyzed the data in an identical fashion for both the condition. So this is how we did. So every voxel from the corpus callosum, the time series was taken, and this was correlated with every voxel in the brain. And so we got 652 images, and then we had 110 subject, and then we did an ICA. And we got resting state network like this, again, which suggests that even the corpus callosum connects with the different resting state network, which allows us to see. And when we just focus on the white matter, we can see these distinct networks, right? So like prefrontal, temporofrontal, occipital, and so on. And this is showing connection between white matter and white matter, these networks, again, showing that each of these networks are uniquely, have unique region, and that there is no there's not much spatial overlap between the different networks, and that these are highly reproducible. This is the map from the first run. This is the map from the second run, highly reproducible. This is the connection between white matter and gray matter. And again, good correspondence and highly reliable, right? Red, red, yellow, yellow, right? It's not identical, but it's highly similar, right? There are some differences here. And then again, this is showing how specific region of the corpus callosum is connected to specific white matter region, right? So showing that this region of the corpus callosum is connected to the visual cortex. This region of the corpus callosum is connected to the frontal region, right? This region, right? So there is a good concordance between the anatomical region for the 2 runs, right? The same regions are associated. Then what we did was we analyzed using diffusion tensor imaging, which is a structural analysis method. And in this case, what we did is, so with diffusion tensor imaging, we can show, okay, this region is connected to this region. And then with resting state, we see that the same region is also connected. The diffusion tensor imaging, to find this, we have to collect an additional run, which we are showing just using the fMRI data. Again, I'm not suggesting we should stop doing diffusion tensor imaging. What I'm saying is that there is a good overlap between diffusion tensor imaging and fMRI analysis of the white matter, and these are the overlap. The red is the overlap. However, not all regions have good overlap, right? So this has good overlap, this has good overlap, maybe this has good overlap, but there are regions like this one or this one where the overlap is not very good. Right? And there are some technical issues which could be, reasons why there is no overlap, because I think here there is a lot of signal dropout in fMRI. which prevented us from doing that. But nonetheless, between resting state connectivity and diffusion tensor imaging, there is a very good overlap between the two. And then with dynamic connectivity also, we are showing these are the white matter networks. And what we are showing here is, this is gray matter. This is the static functional connectivity between different region. This is the dynamic connectivity. And this is the ICC. The greater the ICC, it's more reliable. So gray matter, white matter, and this is gray matter and white matter. And again, the point is that the range of correlation that you see is typically similar on the same order as what is seen. Like initially, I had thought that I would see that much stronger correlation between gray matter region than in white matter. That doesn't seem to be the case. While gray matter is slightly greater, white matter, the connectivity pattern is still there. And this is again showing the relationship between dynamic connectivity and static connectivity. And this is just showing how if you have highly dynamic, a region where there's a lot of variation in the connectivity, it may simply mean that static connectivity may not be very high, and a region, which has very strong static connectivity, the dynamic connectivity may not be very high, right? And similarly, for white matter and gray matter, white matter, the connectivity pattern are very similar. Again, suggesting that although the white matter signal to noise ratio is much lower than gray matter, they have similar underlying signals, and therefore the pattern that we see is so similar. Right? I think that is all the slides I had. So if anyone, I'll just stop here. So if anyone has any other questions, I'll try to answer.
Thank you so much, sir. So indeed, it was a quite informative and a value addition PPT. And maybe participants, if you have any questions, we can ask, sir.
So in the time someone asked, I should say that I'm looking for a postdoc. If someone is finishing their PhD in like technical areas, please send me an email.
Definitely, sir, definitely.
And it would be to work on white matter, some of the work that I showed you.
Okay. So in the meantime, participants are thinking upon the questions. We already have one question in the chat box by Chayan.
Okay.
Should I read it on his behalf?
Actually,
sir, I can.
Actually,
yeah, I can.
Oh, great.
Hello, sir. Thanks for the presentation. Very informative. So I had a quick question that, like, why the brain is fundamentally so active at resting state? As you nicely explained, that the resting state activation is kind of a superset of the task-based activation. So is there any hypothesis or any work on understanding what is the purpose of this resting state activation and networks?
Right. That is a really fundamental question that many of us are trying to answer, and maybe I could give it in very simple terms, and then like based on other questions. So maybe the simplest way is that if you have a garden, right? Even when you are not growing vegetables or planting flowers, you still have to take care of the garden, right? And I feel that in the brain, even during rest, um, the different functional regions still needs the blood supply. It still needs the oxygen and other um, factors. Otherwise, the tissue is going to die, like in stroke. So then, what is the best way in which the blood supply and oxygenation is done? And that is where I feel that the different ways, the complex vascular structure and the neuronal firing which occurs work, together, and that the metabolism, um, underlying metabolism that is needed has to be supplied by the blood flow. And that is why there is this waxing and waning phenomena, or the low frequency fluctuations that we see, which maintains like a balance between the two. And that is, I think that is one of the reasons why we have this. Maybe another way is that, like people who have laptops perhaps can can understand, is that if you have a laptop and you, like, when you are not using it, technically you're supposed to power it down, right? But a lot of us don't do that. We just put it on sleep mode, so that there is still some power consumption and so on, which we are willing to tolerate, because in return, what we get is anytime we want to use something, we immediately can, within a few seconds, we can get onto our laptop, right? So in terms of, let's say, visual task or sensory motor system or other default mode regions, all these things may not be active all the time, but they're never completely at rest or completely shut down, except in case of sleep or anesthesia. And what we have shown is that in both the cases, the connectivity pattern is significantly affected. So I hope I answered your question.
Yes, sir. Thank you, sir. And sir, is there any work that is towards confirming these ideas or hypotheses?
Um, so this this idea is obviously in humans, it's not very easy to be able to shut off the blood flow, right? What we can do is the other way, right? We can perhaps give different kinds of anesthesia and see what is happening. But again, and that is the challenge, right? In in any kind of task that is done or any anesthesia that is done, it is virtually impossible to shut one without affecting the other. That is, I don't know of any drug which has no effect, has simply an effect on blood flow without affecting the metabolism. And similarly, there is no drug that I know which will only shut down the metabolism without affecting the blood flow. So, so we have to, we cannot change one without affecting the other. That is the challenge. There's been some work done on using optogenetics and so on, where you can have, you can trigger place the optogenetics in a specific region and then trigger that and see what happens. And people have found similar that, when you activate optogenetics, then those regions, there's more firing and so on. But can it be generalized for the entire brain? I think not yet. But in animal models, I would say there's been more work where people have almost like titrated using different anesthesia and see how it changes. They have also been able to induce stroke and see what happens. So some of those studies have been done.
Okay, thank you, sir. Thanks a lot.
So any other questions from the participants? So I think we don't have any other questions. Actually, mostly our participants, they are the PhD and the MSR students. Actually, they were quite enthusiastic to hear from you. So my apologies, sir, if we have taken some of your extra time today.
No problem.
So thank you so much, sir, for joining us today. And I'm quite hopeful that we'll have many opportunities in future in which we can hear from you and have a more elaborative discussion. Thank you so much. Thank you from our entire team, sir.
Thank you. Have a nice day. Bye.
So thanks everyone for joining us today for this wonderful session. So we'll catch up again tomorrow at 5 o'clock for the session by Professor Chinmoy De. So we'll meet again tomorrow. Till then, please take care and have a nice day.